السلام عليكم ورحمة الله وبركاته. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقه قولي. Assalamu alaikum wa rahmatullahi wa barakatuh, everyone. Brothers and sisters in Islam, I hope everything is well with you all. Inshallah ta'ala, we'll be going through the BIOC17 Uncle and Lab week 11 review of weeks 1 to 5 bid'Allah ta'ala. So bear in mind, inshallah, we'll have students inside, and inshallah ta'ala, if you're coming in thereafter to listen to the recording, feel free to share your comments bid'Allah ta'ala as well.
Good morning, Mohammed.
Good morning, Dion.
How are you?
I'm great. How are you? That's great. So we'll let people come in. I wanted to start around 10:10, just in case, mimicking our labs together. Um, in the sense that we start 10 minutes after. Overall, how's everyone doing? Don't worry, I don't mind if you have questions or if you have thoughts to share in the meantime. I'm doing good. Yes, and also likewise, I forgot to mention, if you need to text in the chat, that's of course 100% okay. Just let me know accordingly if you have questions. I'm doing great, Laiva. So hopefully we'll get people from the practicals to just hopefully get some um, practical advice and content revisions or some understanding about, I guess, to prep ourselves, all of us, including myself, for your lab exam, which is going to be on the 23rd. So that is, I believe, 2 or 3, technically 2 weeks from now, Wednesday. So about 15 days from now. Welcome everyone who's come in, and we'll begin at 10:10. 10:10. Also, if you have friends, for example, in our practicals or from other practicals that may have maybe aren't aware that we're still having this review, feel free to let them know that they can come in. May be beneficial, and I just think it's good to have like an extra or perhaps a bonus opportunity for understanding the content in prep for the final. So hopefully that's okay. And if you have friends, feel free to just let them know. They could gain access to the same Zoom link, which I'll also share shortly in case you guys need access. Oh, okay. I can try to allow you guys to rename. Let me just see what features adjust. Now you should get access. Let's see. In case you need to rename your name on Zoom, no problem. Also, as a premise, I'm just going to say that I'm primarily just going to go through the content review, but also try to incorporate like some connections and perhaps integrate topics that maybe you would have probably have not considered, or perhaps as well clarify some points that maybe we weren't able to touch on during the practicals together, or if perhaps there are things that could have been adjusted accordingly from the content, so that way you guys will be able to also understand certain topics more easily. And similarly, feel free to participate, ask questions. The format will be pretty much 25 to 30 minutes, maybe 35 minutes per lecture. And then hopefully we'll have, inshallah, about 15, 20 minutes at the end for Q&A, where to go over some pressing topics or some questions that you may have of concern. Okay, any questions, by the way? Just a brief disclaimer, I may not be able to see questions until probably the end of each seminar, so or portion of today's lab review. So hopefully, if you guys have questions, feel free to let me know. I'll be more than happy to help. And I also have a timer, so hopefully we'll go through the material with ease. I also want to go cross-check the lab manual with you guys, so that way, if there are things that I've highlighted in the manual that we may have not touched, we can briefly go over this as well. So we'll try to go over material about 1 to 2 minutes per slide, and then the remaining portion, or even a minute less per page of the manual for the given exercises. So, okay, so from lab 1, right? You know my name, hopefully, with the email muhammad.saeed@utoronto.ca if you have any questions. We went over light microscopy, and pretty much as you could see from the outline, we have a set of techniques, simple staining, hanging drop, and motility test, as well as the negative stain and flagellar stain. Lab rules, as you are aware, not in the lab right now, but this is primarily introductory material. And similarly for light microscopy, you generally want to know like the different anatomical components of a light microscope, even in general, especially the topics that are, or especially key items that perhaps are noteworthy for specific components of visualization when there's errors or when you need to troubleshoot your microscope. So for example, the diaphragm, sometimes it's too wide or too closed. Sometimes you haven't focused well your objective lenses from the fine to the coarse adjustment knob. Normally when you're going from 4 to 10x objective in the lenses, you're adjusting the coarse adjustment knob to move the stage up and down. Whereas for the fine adjustment knob, you still are doing this, but at minuscule levels or degrees. That's how you're able to hopefully capture the resolution to the best of your ability at lower objective lenses. And the same applies for 40x and 100x. However, when it comes to at 100x, the special tip that you're using often, and even at 40x for some microscopes, you are using immersion oil. Does anyone know why we use immersion oil? And forgive me if I'm too quick, by the way. I just want to budget time. Like, I can slow down if needed. Indeed.
I think it's to sorry, I think it's to reflect light. It's not to increase magnification.
Reflect light. Okay.
Or allow
Sorry.
it to go in better. Sorry, I'm not explaining this well.
No, I think you're doing it well actually. So essentially that's correct. You are essentially limiting the diffraction. Sorry, the diffraction in the sense that you have so when you have light rays passing through your light source, be it at the bottom or for inverted microscopes at the top, as your source of light. So let's go back here. The idea is that you have your light source underneath for these typical light microscopes. Well, for inverted microscopes, you have them on top. But the idea is that for microscopes in general, you have your light source passing through your slide, and then there through your specimen of interest. Usually you have a cover slip that's above your specimen of interest, and thereafter you have light refraction, you have diffraction, and other paths or patterns in which light passes through and the light rays in particular pass through your specimen. But the idea is that when we've talked about refraction and refractive indices, you have air at refraction index of 1 and then oil at 1.54. The idea is that when you compare these two, the idea is that you're having a reduction in the amount of refraction. And I know I may have said diffraction, but bear with me in that you're reducing the amount of refraction, which you could see can be compared with a wide refraction index or wide refractive range of rays passing through your cover slip, right? Following that, you see a large dispersion of light rays, whereas with the inclusion of immersion oil, you actually have a finer focus in the sense that like the light rays pass through more closely together through your microscopic objective lens, and that hopefully inshallah reduces the amount of not only dispersion of light, but reduces refraction and increases resolution of your species or your specimen of interest, which in our case is mainly bacteria. Does that make sense, by the way? Feel free to put emojis, feel free to say yes or no. Um, This is like a topic you definitely want to know, essentially. Whether or not it's mentioned in the exams, it's very important for other microscopic courses that you may take in the future. Also, if you see sometimes like a black box come into the slide, don't worry, it's primarily like I'm checking the chat, which is why it also appears. But as we continue, something that you may have noticed from the discussion question on the right is that when you're adjusting, and even as you recall from our lab, our first lab together, you would notice that you normally have your 40x and 100x positioned like a Y, and then you would have inserted your immersion oil thereafter on the slide wherein your bacteria is underneath. And so the idea is that when you have immersion oil added, you'll notice a quite drastic change in resolution or sharpness of your bacteria. And this is the same for other cells. So it integrates to like other advanced microscopy courses, even in grad school. If you take research courses, or even if you do research, you'll come to appreciate that resolution is very important, not only for publication, but for visualizing different species of cells, and even subcellularly, like looking at organelles and other different properties of cells. So definitely it's something that you want to bear in mind, especially when if you recall, like from early courses, maybe even AO2, which I teach, that you would notice that, you want to take detailed annotations of certain organisms, be it isopods, Daphnia, beans, or like pollen greens. And the same applies for other cell types, even plants and animals included. So you'll see the connections, hopefully. But then as we went to exercise 2, we went over calibration, and this is something I want to make sure you guys understand, just for context. So especially when it comes to like this table here, maybe a bit like you may have got a bit confused, because you're noticing that you have slash units, so the number of stage divisions. When you have like when you're trying to calibrate your microscope from the very beginning to accurately determine size of specimens, within like either cellularly or subcellularly, i.e., on the surface level, like seeing the whole cell size, or looking down within a cell into their organelles, for example. You want to be able to first start off with your ocular micrometer. So you have your eyepiece, your ocular or eyepiece micrometer. You'll notice on the left-hand side, normally you'll see a ruler, and that ruler is usually just showing numbers, but they are essentially arbitrary, i.e., you don't know the actual length of those OUs or ocular units in between, right? So we have 100 ocular units shown here. The eyepiece or our ocular micrometer would be this ruler on the top. And then you would have had from the very beginning, and I know we share this between groups, a stage micrometer. And that would have been like a defined ruler, but it doesn't show you essentially the numbers like you see here. And so the idea is that for this large stage micrometer, there are fixed units that exist. And essentially the entire micrometer, i.e. the entire ruler, is 1 milliliter, or 1 millimeter, sorry, and then it's divided into 100. So pretty much you have 10 microns for each small increment, which is shown here as 0.01 millimeters. And so when you look at that, right, this is 0.1, this is 0.01 millimeters, or 10 microns, or 10 micrometers. The idea is if you go up to 10, that'd be 100 micrometers, or 100 microns. And so you consider that because essentially when you overlap, and this is where calibrations come in, which we'll see here, when you overlap this value, you'll notice, let's say, if I want to be accurate and actually these table, like these values won't necessarily reflect the values shown here, but the idea is that you would have essentially gone with overlapping the two rulers together to determine the actual size. So I know that this is 15 OUs, right? But then this is 100 100 microns, because it has 10 divisions So there's 10 divisions, 10 small ones. Each small division is 10 microns, so 10 times 10 is 100 microns. And so 15 OU divided by 100, 15 OUs equal 100 micrometers, meaning that if you divide 15 by 15 on both sides, then you would have 1 OU is equal to 100 divided by 15. That gives you about 6.7 OUs, 6.7 microns, meaning that if you were to look at this, right, this small speck, which is 2 OUs, then you would have multiplied that value that I mentioned earlier, which actually you see here as well, which is nice. The idea is that you would have multiplied 6.67 by 2, and that gives you about 3.33, 13.3 microns. Does that make sense? Just to get an idea. Yeah. Okay, this is a great question. From Fatima, does the ocular micrometer at 30 or 60 always have to align with a longer, or does it depend? It can be either or. It doesn't necessarily have to be. So remember that these small ones, you also know, and they're 10 microns, or 0.01 millimeters, meaning that you could technically do it at, so long as like, well, and normally it is the case, as you could see here, that you have the larger stage division matching up or aligning with the ocular micrometer. But technically, let's say these two aligned, and you, like I don't want to waste your time, but let's say if I found another one that actually does align, that is at a lower at a lower value, you would come to appreciate that essentially you could do the same. So technically, let's say this is 10, right? But let's say 11 might OUs matched up well as well. In fact, I'm going to look at another example. If I could see from the units, 6.67. Um, okay, let's say for example here, um, I mean, it's difficult to see, but let's say this is one, we know this is one stage unit. Maybe we'll do two just to make it easier. This is um, 20 microns, right? Each one is 10 microns, so this is 20 microns, and this goes up to about 3 in the eyepiece, meaning that you're going to do 20 divided by 3, so because this is 3 OUs, so 20 divided by 3 is 6.67, which is pretty much the same value. It is hard to see, um, but I hope that makes sense. Does that make sense? Like you could do other defined values. It may not necessarily be this, like the larger stage divisions, but I hope that makes sense. Yeah, okay. And then this is where I wanted to clarify, just to briefly mention that because these are the small stage divisions, which means that, this is 250 of them, which is more than the units that we see here, you would simply multiply these values by 10. So the values that you see here would be multiplied by 10 to get the total value or the total size of your, let's say, specimen in microns. So this would be 2,500. This would be divided, and then this is actually 100. Whereas the ones that are on this side, to complete the rest of the table, you would divide by 10, right? So what would this value be? Anyone know? Maybe I'll type it in. 60, and then this would be divided by 10 as well, so that would be 3. And then just so you guys understand that, then the next step is that for your stage micrometer, dividing it by the ocular, because these two technically should overlap in units, then you would do essentially 2,500 divided by 40. And this is, by the way, at the scanning objective lens. Do we know, by the way, what the scanning is? Is it 4x, 10x? Anyone know? Thorax, yes, that's right, Hajra. So exactly. So you want to keep that in mind, and just bearing in mind that as you, even though these are defined, right? So that it's interesting in the sense that the eyepiece will essentially remain stagnant as you move between, objective lens because it's connected to your ocular lens, which is a separate component, meaning that essentially, as you go from one objective or one mag to another, your eyepiece or ocular micrometer stays the same, right, in terms of like its display, but the stage micrometer, because you're zooming in more, would thereby appear larger in each stage division. So that's where essentially these values would actually decrease, as you could see quite promisingly here. So that's where you want to bear those details in mind. As you can see, it goes from 40 to 16 to 12. But then, like, and how I was able to determine calibration is actually multiplying, or sorry, dividing these two up. And so the goal is I would like you guys to actually do the same for yourselves. I'm just seeing, I think we're a bit close or tight on time. Essentially, aseptic technique, though, is something that you would have been hopefully aware of, trying to prevent any forms of contamination, not just of the microbes between each other and between your bench tops, but also for yourself, right? Protecting yourself from from getting harmed, and also from harming other bacteria, or like essentially disposing foreign antigens or bacteria that perhaps could actually, um, foreign bacteria that could actually grow or essentially deposit onto your plates, onto your tubes, etc. And definitely that's something that, as you can imagine, from our many labs together, you would not want to occur readily. So that's a simple point, and simply moving on to simple staining, it's very simple, as you can imagine. If we go briefly, I want to just go straight to this page in the manual, page 22. You would have noticed that you have the difference between solid and liquid media, but also in terms of your smears, right? So some bacteria grew on solid agar and some were found in liquid medium. And so in either case, you want to make sure that you're inoculating correctly, right, and aseptically as well. So for solid, because you're having, your bacteria already grown on solid agar, you want to actually have it essentially diluted and to actually become essentially adherent to your slide with water, right? So you'd have water added in, a drop of water, you would have inoculated accordingly. For the liquid medium, you would have just taken an inoculant loop and just added in at the 10, I believe 10 microliter edge. Normally you would have just added in what you see in that loop full, and then you would have just smeared it, spreaded it thinly and evenly such that it's dispersed quite readily. Think of it as like a nice medium layer pancake, right? So this pancake is now of your bacteria, and you would have made them circular. So that way there isn't that risk of, um, over over inoculating, sorry, over um, essentially overcrowding of your bacteria in a given area. So the benefit is that you're essentially spreading it out evenly, and you're reducing the risk of inconclusive data being reached. And then you have heat fixing, which I'm, by the way, I have components highlighted here. It may be a bit difficult to go through, but the idea is that you have specific details for heat fixation, the killing of bacteria, coagulation, and the benefits of adhering your cells, as mentioned earlier. And so you want to bear that in mind, as well as making the appropriate mixture with the right consistency, like skim milk. And then one thing that I haven't mentioned, but something to keep in mind, is that when you're flowing down the water, or your decolorizer or your stains, that you appropriately, for the right amount of time, essentially stain this way, but decolorize the other way. Actually, you could even stain both this way. Do you guys know why you would normally want to essentially have the medium or stain of interest pass through this way rather than the other way?
So that it doesn't go into the other drop and cross-contaminate?
Yes, yes, and that's something that may be a bit odd to consider because you technically would have already heat fixed the bacteria. But the idea is that because we're dealing now at the micro level, we don't want to even take the risk of, let's say, a positive capsule, like a encapsulated bacteria passing through hypothetically into what merged in your other smear that would perhaps be your unknown or negative control. So the idea is that you want to bear in mind potential limitations in that regard. And then you have the negative stain, which is briefly touched upon, but essentially this is an acidic stain, which as you could see is for nigrosine. And I'm just going to go back to the slide to see if we're good on time. Um, so maybe let's continue. The idea is unlike simple stains where you have like crystal violet, methylene blue, for example, you would actually have the background typically darker than the stain of the bacteria, which in our case is clear. So it's kind of like an X-ray, right? So you're X-raying your bacteria, right? You're doing you could be doing an MRI, for example. But the idea is that you have your bacteria appearing more white or clear with a greater contrast compared to the background, which is usually dark. And that's the benefit of using like this acidic stain because, sorry, you would essentially maintain the absor your visualization and the morphology or the sanctity, if you will, or stability of your bacteria without having to run the risk of heat fixation because heat fixation actually swirls or essentially um, makes or runs the risk of de-integrating the cell wall or reduces the integrity of your cell, your bacterial cell in this case. And that's why it's often advised in this case for your negative stain to avoid heat fixation. So you want to keep those details in mind. And then you would have normally observed the morphology, right? We could see clusters of cells, right? And perhaps in this case, which is separate from the ones here, you'll notice that these appear like cocci, right? So it kind of give you a hint, if you recall from your unknown assignment, or as we look back into the past, what types of morphology you would expect to see here. But then thereafter, you had the hanging drop, right? And then you have the flagellar stain. Flagellar stain, the different terms are highlighted in the lab manual. So that's something you want to keep in mind, especially knowing the differences between the number and location of or arrangement of your flagella if there are more than one. And then you had a motility test, which you would see in the following slide. But briefly, hanging drop, which was the last major exercise, was, or semi-last, was with your hanging drop, you would have normally had your cover slip. If it was solid, you would have added a drop of water and then placed your bacteria that would have at least reached a third or a quarter of your loop, and then you would have dabbled it around or diluted it into your medium, and thereafter you would have actually placed petroleum jelly, inverted it onto your depression slide, and then re-invert so that way you could essentially see the hanging of your drop of bacteria. And the benefit of that is that at lower to higher mags, you would be able to see at the edge of the drop the morphology or the motility of rather your bacteria. So some are showing true motility, right? That they move in a specific mode of direction, or like for context, they do show an indication of motility, whereas Browninian motion is usually vibratory and they more like stream. So they stream down, let's say, a particular motion of water molecules, right? In the case of your solid agar or even in liquid, they would just move in a stream-like fashion. Whereas for true motility, there is a change in direction that exists typically, and that's something that you would notice between and across different bacteria. That pretty much ends my brief take on, sorry, my brief component on lab 1. And then if we go to lab 2, are there any questions before I move to lab 2, by the way? No questions? Let's continue then. On to the unknown. So in this case, well, your bacteria had 6 of them, and something I highlighted from our labs is subculturing. So I want you guys to understand this. When you're plating cells, right? You're subculturing often, or if you have them, let's say, already in a present tube or in another plate, and you want to isolate them, right? This integrates with week 3, I believe, where you're isolating specific colonies. The idea is that you want to make a pure culture of a specific species of interest, right? So it could be Micrococcus luteus, but not Micrococcus roseus, right? So that would be you trying to take and subculture a mixed culture to actually extract and get a pure culture, let's say Micrococcus luteus. So essentially, subculturing is like you going from one culture, like taking bacteria from one culture to another. And we've done this for tubes, but we've also done this for plates, if you recall the antimicrobial susceptibility test. Give me one moment, folks. Sorry. All right. And so you would have done this essentially to inoculate your bacteria, and this was aimed to essentially prevent the risk of any form of contamination, right? We're calling the aseptic technique, reducing any form of cross-contamination of your previously unknown bacteria from the first stock slant that you had. Normally, and then so after that, you would have noticed that there's a specific trend, right? When you subculture and even when you inoculate, you'll notice the streaking pattern, which maximizes the abundance of bacteria that are able to grow within a given vicinity because they have nutrients from other sides that they could extract and grow upon. But then essentially you want to bear in mind that when you normally try to, get a confluent amount of bacteria, what we've done from our latter weeks is add in saline water into your bacteria, into your bacterial tubes, and then you would have made it more confluent, like in the case of the antimicrobial susceptibility test plates and other plates as well. But then you would have gone here, you would have made a detailed flow chart, right? Comparing the different components, as you recall. There are a lot of them shown in the lab manual, and I would recommend like trying to understand how you would differentiate certain bacteria in the future. But it's not because you've already done this experiment or this as an assignment, you wouldn't you would want to know the main topics, but you wouldn't have to go too trivially down to the nitty-gritty. But try to understand the major components of lab exercises altogether. Now, when you look at gram staining, I hope that makes sense, by the way. When you look at gram staining, if you saw from the lab manual, would have noticed that it's a differential stain, right? That connects us also to our last week practicals, right? Because you would have seen this for the biofilm. Staphylococcus epidermidis is gram positive, as you recall. And I do want to say I would have loved to actually have this more discussion based, but I realize timing is quite short, so we'll keep discussions till the end. That's my goal, and then we can go back when needed, and we can extend the timing also if required. But I hope that makes sense as well, because it's honestly to describe all the data, to go through it with you guys, it does take time, and I know that that's why the reviews are typically condensed, but hopefully when we inshallah go through the questions, certain topics that you want to go over more in depth, we can go through them, even including the manual if needed. But briefly, let me ask, do you guys know what the mordant is? Or what function it serves? Because this will actually help us understand how you're able to differentiate gram-positive versus gram-negative cells. And you could talk, you could put it in the chat, I don't mind. That's right. That is right. So, Vasnavi pointed out that it helps the bacteria hold on to the primary state. And for context, it's typically iodine, right? And the idea is that coupled with crystal violet, so think about it like this. Imagine yourself. And for bacteria, at least, if they're gram-positive, they have a thicker peptidoglycan layer or wall. And essentially, you'll notice that as the crystal violet goes in, it's a small molecule, yes, but essentially, it actually is like the nano range, like you would actually technically say it's a medium-sized molecule that essentially, as a dye, goes into your all bacteria, gram-positive and negative. But then when the mordant comes in, you have essentially the with the crystal violet, the what do you call it, the complex form essentially within, and what makes it noteworthy is that for thicker peptoglycan layers, it is retained essentially with the crystal violet. So imagine you have your wall, thick wall for gram-positive bacteria. Crystal violet seeps through. Same with your mordant. But when the mordant and crystal violet come together, they become a larger complex, right? So now they're stuck within your thick layered peptoglycan of your gram-positive bacteria. If it was hypothetically less, like if it was thinner, like in gram-negative bacteria, when you decolorize, right, with 95% ethanol, it would seep throughout, right? So the complex is actually able to come out of your of your of your bacteria, whereas for gram-positive bacteria, it would remain and be retained within the bacteria themselves. And so what you can imagine then is that following decolorization, you actually have dark purple appearing bacteria, but colorless gram-negative bacteria, because essentially what you thereafter do is add the secondary stain or the counter stain, which we know is safranin, right? So that's where you essentially have safranin coming in and then seeping throughout from the gram-positive bacteria, but being retained for gram-negative, right? Technically, for gram-positive, it's already stained with the dye dark purple. So you typically, with that clear dark contrast, you wouldn't really have the counter stain come in, or at least be retained in the bacteria themselves. So essentially, following the rinse with water sideways of your stain with your slide, you would essentially have gram-positive appearing like this and gram-negative appearing like that. So gram-positive purple, gram-negative pink. There is a concern of gram variability, which I think, let's see if I have time to share that now. I think I do. So if we look at the lab manual, you'll notice there's certain, let's see if I should save it. I want to see if I could save ourselves time. Maybe I'll save it and then we'll go through the lab manual all together at the very end with questions, so that way I can answer them more directly for you guys. But the capsule stain is another type of stain, which is actually differential. It also has like, well, if you think about it, you have like the structural component that is like depicted, right? Because it is this particular form of glycocalyx, which I mentioned, by the way, for biofilms, because it's actually is connected from our week 10 results, lab 2, like the final exercise, one of the semi-final exercises of the lab, because the idea is that you would have seen a dark purple coloration Streptococcus mutans, for example, is gram-positive and capsule staining. And so the idea is that if it's encapsulated, you would have this glycocalyx. And glycocalyx is like a layer of sugar, a waxy layer, but it's composed of polysaccharides and of protein, typically, and that helps to not only prevent desiccation, but also helps to grow communities. Wow, I didn't even know it. It just showed it here, biofilm formation. So that's even perfect. So you guys can actually, wow, that's amazing. You'll see how it really directly connects to the material that we've touched upon. Wow, I didn't realize that. See, you can imagine how many things integrate, as I just mentioned, right? So hopefully that's something you can appreciate indeed. And I'll just have this shown a bit higher. But the idea is that this connects to the following setups because you have two differential stains. You have your Congo red, right? Which is typically differential, right? Showcasing a reddish hue. And then you have like a higher pH, and then acidic blue hue at a lower pH. The idea is that following these two stains, Congo red, which essentially would go to the formation of your red background, you'd have the Manival stain. And this is something I want to bear in mind so you guys understand it. It is basic, right? So this, like the Camelfusin dye that's mentioned in the manual, you'll notice that it forms this reddish hue, essentially dark red hue, that essentially you want to keep in mind is similar to a degree to what I've mentioned before in the sense that these capsules will essentially appear darkish, or sorry, I guess like light red with the Congo red, essentially. And I don't know if it's shown here. I thought it was, but let's see. Yeah, perfect. So it's shown here. The idea is that you would essentially use the Congo red, blot it accordingly onto your slide. Then you would have used what you've done for negative staining, some of you at least, for essentially spreading your bacteria from one edge to another. And then on the faint areas is where you typically would do visualization following the addition of Manival stain. So it's important to bear in mind the different functions they have. But essentially, the capsules will not interact with either Congo red or the Manival stain, but will remain halo. So like you want to think of it like this. You have, oh, sorry. Sorry about that, folks. Pretty much, I believe what I've said thus far. Yes, so essentially, you have two different components to bear in mind. So you have the acetic acid from the fusion basic dye, which is it's a bit separate, but essentially the idea is that the function does lead to the lowering of your pH, which causes the Congo red to appear in the background as grayish blue. So just think of it like this. Congo red goes in for the essentially addition of for to denote your capsule stain. It will go in as Congo red inside, and then essentially you have the Manival stain that appears on the as a contrast of your background outside, and then the halo, which would be in between for encapsulated bacteria, would be shown essentially as a clear white capsule. It's a bit difficult to describe in depth or at length for the shortness of time I have, but I hope that you guys are able to understand that why it's important to bear in mind not just the different components that we have, Congo red being, the acidic stain, and then the acid fusion Manival stain being generally a mixture of acid and base, which is quite ironic, but essentially they serve different components to essentially have the stain either encapsulated retained for encapsulated bacteria, and then the Manival outer acidic fusion, if I'm not mistaken. sorry, the acidic acid will thereafter form the background um, as the darkish blue hue. And so essentially the important step is that you bear in mind that you've added in sufficiently the amount of both components, both stains, but also making sure that you would have not heat fixed, because otherwise the capsule can actually disintegrate, and also you run the risk of distorting the capsule itself in morphology. So I hope that makes sense. But essentially, you have some good examples here. This is not capsulated. This is, without looking at like the comments, you can see clearly that this is capsulated. This appears not necessarily. Like you could see the dark purplish hue. But one thing that I think, and we'll talk about this actually with the lab manual with the details I provided, but you then generally see if you have a close look. Actually, it's very hard to see for panel D that it's positive, but you could see like here, you see some bacteria. You see the hue of like the caption of the red, essentially the red coloration here, 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 and here for darkish purple hue. But essentially for different bacteria, you may even use similar but different dyes that essentially work in a similar way for capsule staining. The idea is that the background is stained, the inside is stained, and if there is a halo, that depicts your capsule. Or if there's a capsule, it would be shown as a halo. And then the motility test. I want to show, well, actually, I'll describe it with the manual shown, I believe, for exercise 4. Let me just go all the way down. It may be hard to see. Yeah, but this is, by the way, what I was referring to for the different flagellar arrangements that you guys do want to know. Even if it's just a single question, it may not even be, but it's good to know the implications with the applications described. And I apologize if may not go the same way as I expected, but generally speaking, you have the differences of morphology that I mentioned for gram staining, gram positive versus gram negative. Bear in mind that you have an outer membrane for gram negative bacteria as well, which hopefully you would have understood for components of UV exposure. And then you have gram variability, which I wanted to mention that could exist if you have two very thick smears that exist. And then cultures, if they've aged, that could also have an impact. And then those are the different conditions highlighted here. If you over or under decolorize the false gram negative versus false gram positive bacteria. But I'm going to just go straight down, and then hopefully when it comes to questions, we'll go over them in due time. Yeah. This is where I wanted to talk about motility test. Briefly, you have TTC. It's a component of this specific 0.4% agar, and the idea is that it serves as an electron acceptor, and following metabolism and migration of bacteria that are motile, then you would expect to see a red coloration throughout your tube. It doesn't mean like you see like that red smear, those are still bacteria, right? It's just non-motile. Like they're still able to metabolize the agar, but they're not able to move beyond your smear or your stab, or like the streak that was shown here through the agar. So meaning they're non-motile. And this is usually more easier to discriminate from the hanging drop, right? Because you only have a finite amount of time to look at the edge of a drop and focus in to see, oh, is it vibrating like brown Indian, or if it's actually moving directionally as true motility. So that pretty much sums up that last component. And then you have the acid fast stain, which I want to go briefly touch upon. And I touched about the fusion dye from before. This is the carbofusion rather for acid fast staining. And then the important point, unlike the capsule stain, is that you are adding heat. There's technically two versions, as you probably recall, but the idea is that you want to do a similar technique of being able to counter stain and stain your bacteria either with the cobalt fusion, the primary stain I mentioned before, and with the methylene blue, which is usually for non-acid fast bacteria. The difference between the two methods that are shown here, the Zal-Nassine method, is essentially you are doing the primary cobalt fusion stain with the addition of heat for a given period of time for 5 minutes, whereas and you'll see like the differences in color here, which is typically shown for histology as well. The difference from that Zile fashion, like the previous method, from here, the Quinoin cold method is that essentially, as you can tell here, you're not adding in heat. And so there's a specific other component of the cobalt fusion that is used with a higher concentration of phenol so that it sticks and penetrates the waxy layer, which if you think about it, think about your hands when you have oil, the phenol, which is able to essentially um, have or in like negatively interact. Sorry. Essentially, think of it like using sanitizer because it's an ethanol or alcohol, right? So it's a phenol. If you have oil and you rub it on from your hands, you have oil, and then you use sanitizer, you're normally going to sweep out quite readily the oil on you. So it's similar in this regard, in that the phenol is able to pass through quite readily the waxy layer of the cell, which is essentially your acid fast component, which is how essentially you're able to readily pass through even though it's for a longer period of time, but without the use of heat. So that could be beneficial, especially if you wanted to see live bacteria without essentially heat fixing. Generally, this could be another mode of essentially observing your bacteria of interest. Does that make sense? Any questions? So think of it like this. Carbonyl fusion is your primary, secondary is your methylene blue, and acid alcohol could be your decolorizer. If we recall, there's also an exception here. Actually, maybe it's mentioned in the following. I think it's mentioned in the following slide, where essentially you use another set of decolorizer, which we'll talk about in due time. But any questions? Sorry. It's a bit high paced, I must say, but I hope you guys are able to understand. And if you over decolorize, actually, if you over decolorize, what do you expect here? Think about this question. You're dealing with mycobacteria tuberculosis, which is, as you could see, relates to a given condition, TB or tuberculosis in the lung. As you can imagine, you perform, let's say, an acid fasting, right? So I want you to imagine this. You could see the purplish red hue, likely indicating that it is acid fast, right? Or think about it for a moment. What do you think would happen if you over decolorize? Is it non-acid fast or is it acid fast? One. And then two, what is the role of the acid alcohol? Like, as you can imagine, it serves as a decolorizer. So if you over decolorize, what impact would that make, essentially? So is it acid fast positive or negative? And the second question is, what impact would over decolorizing make? Like on your slide, you expect the color to change? To what color? Think about that. And feel free to mention it.
It's acid fast positive, and probably over decolorizing, you'll probably get a false negative because you'll over decolorize and you'll lose the stain.
That's right. So to now one point though, is it gonna appear then purple or blue? Like reddish purple or more tainted blue?
More tinted blue?
More tainted blue. So, and that's correct for both sides. Now, to answer for Fatima, it would appear negative, and that would be correct. But technically, it is actually positive. So that's why it's very important that you don't over decolorize or under decolorize. Normally, when you over decolorize, you run the risk of getting false negatives, i.e., it appears negative, but actually is positive. But if you under decolorize, i.e., you added less, or like let's say it was a 5% concentration of acidic alcohol, if you added in 3% accidentally, or you added in a lower volume, then technically you could expect that you would see like, and I want you, when I say lower volume, I'm saying like, let's say hypothetically you have a big smear, but you only added it to like one portion of that smear, then technically what would happen is that for the other remaining portions, if it indeed essentially wasn't decolorized or sufficiently decolorized, you'd have essentially false positives, meaning that it actually looks positive, but it isn't. Uh, no problem, Cressy. Who's Cressy, by the way? I don't know who Cressy is. Maybe some of you know. That's okay. But hopefully that person enjoyed it. Now, as you go to week, by the way, any other questions? We are good for timing, but I want to spend a good amount of time, hopefully, with looking through the lab, like the highlights I've touched upon. Um, let's go to, in the next minute or so, serial dilution. Okay, so, and this appears more simpler, but have a look at Appendix A. Right, and that's when you would have, submitted your unknown assignment, flow chart, data sheet, as we talked about earlier as well recently. But essentially would have, and that's where, it's very important to highlight the importance of having like an even pancake, right? You don't want it to have like many ripples. Sometimes ripples are good actually because it shows to me that you've tried your best to essentially even out as much as possible. Yeah, within a wide smear, your solid, let's say your bacteria grown on solid agar. But the idea is that you want to have it quite uniform, ovoid or circular normally, and you would have had the staining done also quite accurately. Um, so let's go with briefly serial dilutions. Um, I think I should go with let's go with the manual because I highlight specific components that I want to clarify with some of you. When you're doing so the you know the purpose, right? The purpose of doing the serial dilution. And the purpose, as you could see, is to determine original population density or cell density, right? I want to highlight something though. When you're determining, so let's say it's viable counts, right? From your plates. You are counting the number of colonies, right? Viable colonies, live bacteria, right? That have grown from CFUs, colony forming units. I say that particularly because I don't want you to assume that you see a bacteria, and maybe we'll talk about it in due time, but the idea is that when you're calculating the density, you're trying to get an estimate of the number, or the total number of bacteria or colony forming units in 1 milliliter of sample. So if you wanted to apply this to real world, let's say like you're doing a particular experiment, let's say of transformation or conjugation that's going to come up in the following uh, component of today's review for Lab 4, you would have come to notice that, there's a specific volume that you're using, not only in mils, but sometimes in microliters, or in down the road, maybe in uh, beyond the milliliters, you may be using nanoliters, right? Or even liters for larger apparatuses. So you want to bear in mind that the volumes, the concentrations, and the densities that you're using are accurate and are transferable, not just as skills, but also as part questions for, let's say, your lab exam. So just bear, and I'm not writing your lab exam, I'm just giving it as an example, just to be able to understand the importance of your units. You're calibrating correctly, as we've done for the ocular micrometer, to determine size, but you also are accurately deriving the appropriate units for further operations. Um, so I hope that makes sense. As you scroll down, you could see the importance of endospores, which you've seen from your UV FLR, which I, alhamdulillah, have completed marking. But essentially, you want to understand the functions of endospores. And something to point out is that they generally remain vegetative and normal conditions, meaning that essentially they're not sporulating. So like the idea is that you can have bacteria like Bacillus subtilis or B subtilis, for example, for short, that is able to grow, right? Normally, just like other bacteria like E. coli, Staphylococcus epidermidis. But the difference is that when there is a need, right, let's say reduced nutrient availability or flux of metabolic waste or high temperatures, low pH, recall the environmental conditions exercises or assignments that we've done in the past with the presentations. The idea is that like with extreme extremities, you have these vegetative cells no longer growing, but you have sporulation existing or occurring, meaning that you have a formation or production of endospores. And those endospores are shown in malachite green here, right, with the different orientations of central to terminal slash subterminal here of circular or ovoid of endospores, but their functions remain once again to persist existence or persist growth in unfavorable or extreme conditions, right? So they're able to essentially be able to survive, and then when pH is reduced from, let's say, 10 to 7, or temperatures go downwards, then you have the ability for these endospores to stimulate germination, and then these cells thereafter return back to their vegetative or normal states. So think of it like ground states. So generally speaking, we have these benefits that have been highlighted here, which connects us, believe it or not, to many exercises and weeks we've done before, be it of thermal death time for high temps, to generation time of changes in temps, to chemicals that have been changed in, like what we've observed for the differences in even, let's say, differential stains, right? Because we've dealt with, different concentrations of NaCl, for example. But you have antimicrobial agents, antimicrobial susceptibility test. Even though we haven't really touched upon B subtilis there, you should be able to consider the impacts that this has, perhaps for your lab exam and perhaps not. But think about it likely for prep of your lab exam to see how you could extend, even if it's not touched upon. Think about the implications of like growth and essentially subculturing, or further analysis of essentially endospore or sporulating bacteria. So think about those details. And what I wanted to highlight for the endospore stain is that we actually used water, right? Hopefully. Because the interesting point is that malachite green, which stains the endospores itself, following the heating essentially, because bear in mind they're heat sensitive, or sorry, heat resistant, so they can be able to grow or be able to live even at high, or that at moderately, like at a essentially over boiling water. I just want to be sensitive because technically speaking, you wouldn't expect to go to the far extremes of like 200 degrees Celsius, but The idea is that, these endospores with their cells themselves could be stained with malachite green and then essentially decolorized with water to thereafter add in saffronin, which we've seen, right, from gram staining as well, and other stains to essentially coat um, um, the internal structures or the cytosol per se of the vegetative cells themselves with red. And then if there are endospores present, then they would appear green, which we saw earlier. I do want to highlight as well, maybe it'll be better with the slides. So let's go back to slides. I want to highlight that, and this is something that may be a bit contentious, but, and this is why I was concerned about saying that these cells, for example, at 48 hours, would essentially be vegetative. Not all of them appear vegetative. As you could see, some of them actually have spores, meaning that they are undergoing sporulation, right? So they're no longer germinating, but they're no longer in their vegetative states, i.e. without endospores. So just think about it like this. At 48 hours, because it does take a period of time for sporulation to occur, the idea is that you would have some spores following, given the certain circumstances, sporulation could occur at 48 hours. At 7 days, however, you could see many of these free endospores would have actually likely not, let alone being shown here outside of bacteria, you would likely actually expect perhaps that if you were to decolorize and actually rinse after, hopefully not, but if you were to rinse or wash the endospores, you may not actually see them, right? I say this, the reason why I make mention of this is that yes, they could be sporulating, right, at 7 days, but you want to know the effect of aging, right? And that also connects to the UV assignment because technically, if you find at 7 days, hypothetically, a difference from 24 hours, then, or even in this case, 48 hours for B subtilis, then you could kind of thus decipher the impact or the state that those bacteria are in, i.e., if they were once with endospores, but now they're no longer with them, they're likely no longer sporulating, meaning that they're likely now back to their vegetative states. But if they had the endospores inside, then they're more like within the bacteria themselves and they're producing this or maintaining the endospore, then likely they are in extreme conditions. So that's where, the impact of UV comes in as well. So keep that in mind. I know it sounds different from what maybe it was described from the past, but it makes hopefully more sense in light of what we've described now. And then pretty much that would have been your exercise, and then serial dilutions. Actually, before I move further, does that make sense? So if let's say just as a question, um, or I don't want to go into trivial conditions, but let's say you have B subtilis at 48 hours, uh, or even 7 days. We'll say 7 days. You assume that it's sporulating, hypothetically. Um, but when you do your slide, right, like you prepped it up, you place it into the water bath. So you first started off with malachite green on the water bath. You would have before then heat fixed, right, your bacteria onto the slide. Thereafter, you would have essentially placed your slide with I believe you would have already heat fixed. So yeah, you would have already prepped your smear. You would have let it air dried or heat fixed. Then you would have placed it onto the malachite green on the water bath, and to maintain, recall, like the heat exposure, right? The humid temperature as you're continuing to add in malachite green. I want you to think if your results at the end following decolorization, actually, let me go over it. You went through following the malachite green, you would have gone through decolorization, right? And then hopefully you would have expected B. subtilis to have green spores, right? Or the presence of endospores within the bacteria. You then after followed through, and this decolorization was, after 5 minutes of adding in malachite green onto the plate, onto the to the not the, like the cover slips, kind of the filter paper you would have added in multiple rounds, right? You recall briefly, but essentially suspending your bacteria with the malachite green in exposure at humid temps. And then thereafter, you would have done decolorization on your bench tops with water, right, with a slide trace. Thereafter, you would have done safranin, added it on top, incubated for about 30 to 35 seconds. And then thereafter that, you would have rinsed again and observed on the microscope. If you let's say, if you saw, like, and I want you to, like, if I were to see your, let's say, slide, and let's say I knew it was supposed to be B. subtilis hypothetically, but for like considering the fact that your positive control and your unknown appear to not have endospores, then what concerns or what limitations could we derive? Think about some like common things that maybe I described, but maybe we didn't highlight as limitations or concerns for consideration. And this actually connects us to the UV-FLR, believe it or not. Because just like how you expose UV at a certain height, you may also want to consider how high the slides are away from the water, right, for humidity and vaporization. So think about that. What are some things you want to keep in mind?
As you said, maybe simply the heat transfer between like your water bath and your slide is not good, and therefore, or your slide just wasn't hot enough to like let the malachite green like soak in. Maybe you weren't using enough malachite green. Possibly like the bacteria itself wasn't like kept in like desolate conditions, or it simply was not sporulating. It was more in a, how do you say, not vegetative, but the conditions were too good. Spores just didn't form.
That is, so everything was correct except for the final part. I think maybe Dr. Brent would contend. Reason being is because you technically would have already had the spores produced by that time of you doing the staining. So, sorry, give me one moment. Okay. Yes, so essentially, as Garvin pointed out, right, the distance of your water bath, even keep in mind the water goes down, right, because it's also evaporating, right, from from the water bath. So even from the beaker itself, just keep in mind that, as groups came in and out, the water is going down slowly and slowly. So the distance and in terms of the effect of essentially maintaining the vaporization or the even the condensation rather of your malachite green onto the surface of your bacteria may be reduced. Feel free, by the way, people. I noticed you guys are mentioning in the chat. Keep doing this. Keep adding in points, and I'll describe them as well. And some other points include, from what I was about to also mention, is the filter paper itself. Some of you, and I know it was a bit hard, but if you had three, let's say, cultures, hypothetically, or even two of them, but they were widely apart, and the filter paper was, let's say, not easily separating both of them, or essentially you had multiple filter papers on top, then that also runs the risk of reduced exposure to malachite green, meaning that there's a reduction in the ability to actually observe those endospores, as the malachite green wasn't able to sufficiently pass through your filter paper directly into your bacteria. But there is also a purpose, right? We use filter paper. Imagine you just added a malachite green on top of your slide, right? You would have had likely background exposure with malachite green, which would have made it more difficult to observe, almost like an endospore stain coupled with a negative stain. in green. So you would have seen a green background. And I say that because you're vaporizing it technically on your slide, so it's kind of sensitive. Bear in mind that, using the malachite green as a strong dye that's going to be absorbed into your endospores that are also resistant themselves, you want to make sure that the filter paper accounts for just essentially absorbing in readily where it is needed, i.e. for your smears of interest. And then you have, yeah, exactly. So both Vasnavi and I believe someone else. Oh, okay, Vasnavi pointed this out. Correct, decolorization is very important. If you didn't add in sufficient amount of water, then you would under-decolorize, meaning that you would see false positives. But in our case, we saw false negatives, perhaps, right, with the control indicative of this. So with that in mind, thus, what you would expect is that if you over-decolorize, i.e., if you spread too much, if you spray too much water, then you run the risk of essentially reducing the visualization of endospores, especially now that you would have thereafter added in safranin, which is another counter stain, so that could have actually filled through the entirety of your bacteria, making the endospores more difficult to see as edges. So some bacteria might have been, yes, and that is a separate point, which many of you have also pointed out for you with the FLR, stationary versus log phase. And that's a good point. At 7 days, this is a good point to highlight, which is interesting though, because our results appeared otherwise. But it is still an important point as a limitation. The control culture may have been, and that's another thing, right? Good point from Diane. Uh, um, because you also want to consider the age of your cultures themselves. Some of us also saw from, I believe, our transformation assignment or transformation exercise for Lab 4, the differences in exposure and the effects, sorry, the thinness or the faintness of your cultures, which would have made it more difficult to observe and thus essentially gain the best or extract the best or the highest yield of bacteria for our exercises. Technically, sorry, it wasn't the transformation. There was one exercise that someone was showing me. It was faint. We had faint cultures. Nonetheless, I think they were for latter weeks, so maybe we'll touch upon them from the lab manual in our next review. But the idea is that you want to bear in mind the conditions, right? Are they too young, too old? Do they have spores or not? Did they already germinate, right? Are they back into favorable conditions, right? Because technically, as we use them, they're likely at 37 degrees. If the at room temperature, meaning that they should likely, if they optimally grow at this temp, they should be able to not they should be able to germinate or perhaps sporulate if it is at their extremities. So you want to bear those details in mind. Be sophisticated, be understanding and cognizant of the different conditions that may be at play. And that's pretty much what we've touched upon. So we're ahead of the game, inshallah ta'ala, to go to lab 4. Any questions? I'll go forward and then we'll go through streak plating, which is a separate technique we've described, and then your plaque assay and the other assays that we've done thereafter. Media prep, understand a bit of it from the appendices, but focus more so on the content that I've described thus far. You notice, as I touched upon, right, pure cultures versus mixed cultures when you're doing subculturing. Streak and spread plating connect in a similar way, in the sense that for spread plating, you normally are doing single colonies, i.e., or like you're growing like for viable counts from the generation time. For example, you're trying to derive, the number of colonies or colony forming units from a single culture, right? Usually indicating or indicative of a pure culture. And then when you're doing streak plating, you're essentially diluting over a period. So for serial dilutions, even though I didn't touch upon it, for serial dilutions are typically doing spread plating thereafter, right? because you're doing multiple dilutions, and from those separate dilutions, you're doing spread plating of each diluted, i.e., from, let's say, the number of bacteria are being diluted across a series of serial dilutions. So you're doing that in multiple plates. Whereas for streak plating, you're technically doing it on a single plate. You see multiple, like streaks of bacteria, right? Streaks of colonies that slowly and slowly become more thinner and disparate until they're separated to nearly individual colonies here. And that's where, you could kind of appreciate that if you wanted to do, let's say, transformation, which is in this week's exercise, then you would be able to separately take out, let's say, HB101 E. coli, and thereafter continue with the PGLO or continue with the exercise, essentially. I hope that makes sense. And then streak plating, streak plating, think of it like, taking in streaks from one edge to another. And this is, panel A is actually an example of like an improper streaking, because essentially you have, yes, technically you do have some individual colonies, but bear in mind that, let's say, for example, and I want you to think of it like this. You have a given mixed broth of M. luteus and E. coli. If you're trying to isolate these specific colonies, would you essentially, even for streak plating, would you do them on the same plate or separate plates? I want you to raise your hand as a participant if you think it would be separate plates. So just to repeat, for panel A, based on the question here, you have two different bacteria, bacterial species, and you want to do streak plating. Would you do both colonies on the same plate or on separate plates? Raise your hand if you think for separate plates. Okay, I have one raised hand from Alta. That's good. Okay. For the same plate, raise your hand. And then Alta can lower her hand. Okay. Oh, I'm seeing only a few people raise their hands. That's a bit concerning, folks. I hope you're paying attention.
I don't know how to raise my hand. I'll say on separate plates.
On separate plates, right? Okay, so interestingly, I'm seeing a mix of both. Feel free to lower your hands. Sorry. I'm not judging. I don't want to like assume otherwise. Maybe some of you had to briefly exit, but bear in mind that when it comes to this, at least, streak plating you generally want to do, there's two conditions. As you could see here, when it comes to the following components, you want to isolate the colonies, right? So if you're isolating them, if you were to do them, technically, you would have to bear in mind if it was on the same plate, you would have had to have them like disparate, i.e., separated. What do you call it? Along a line. So, like, if I hypothetically was to do streak plating, it is more laborious. It could be done, but it's not the most favorable, essentially um, method. The idea is that if you wanted to, theoretically, you could do streak plating from one region, bearing in mind that you go from one quadrant to the next, right? So this probably would have been filled with colonies, the smear, but then you would just briefly touch a smaller region. And then likely from here, you would have had them more disparate. And then I could actually go from that end to eventually have the remaining portion essentially served as or diluted across your agar plate from the media itself, diluted to eventually individual colonies, all for one, let's say, one bacteria, which in this case is E. coli, and then separately for Micrococcus luteus. However, if you were to do it like this, the problem is that, okay, you can see technically separate bacteria, isolated colonies, but you wouldn't really know unless you knew, like, differentially, let's say for Staphylococcus epidermidis and E. coli, the differences in shape and morphology and briefness and color. But If you wanted to be more reliable, it would be, and you can technically do the same on both sides, like separate for E. coli, separate for Amludius, knowing the labels, right? But if you wanted to be more accurate, you would technically do them in both separate plates. You could do it in both plates, but in a single plate, it takes more time to essentially. It would be something that you want to be you can grow in the like in rigor essentially in being able to understand. But typically, um, if you wanted to do the next step, which is the final question here, you would essentially subculture into separate plates, either for spread plating, right? Like with essentially spreading with the L rod across your plate of interest with your bacteria, usually zero point 0.1 mils, or with streak plating to derive a greater dilution of spread plating, so a dilution of your bacteria across a single plate. So I hope that makes sense. Um, just gonna, I want to make sure you guys could see the following slides. Yeah, give me one moment. Any questions? So, like cereal, oh yeah, so we already talked about this. For cereal, and well, something to point out, I mentioned about the briefly the functions for serial dilution. By you essentially being able to isolate or to derive thus the original cell density of your bacteria or sample of interest, right? And so you're doing this at serial dilutions. Serial is like repetitive in this regard. So you're like doing this at numerous iterations. You're repeating, you're starting off with a stock or another tube, and you're going from 1 mils of that tube to 9 mils in that diluent. Could be water, saline, etc. And then you're just repeating that of taking 1 mils from there. But bear in mind that, okay, after you place 1 mil from the stock, let's say into 9 mils here, you have a dilution, right? That dilution increases, the factor increases each time that you serially dilute your inoculant, right? So, or your bacteria or cells of interest. So you repeat that step or that stage-wise repetition or inoculation of bacteria from your saline into separate tubes. Or sorry, your diluent into separate tubes. And then the idea is that you plate thereafter, spread plating the different bacteria across from each tube, 0 0.1 mils, into the following sets of plates. I'm just gonna, and does that make sense, by the way, folks? Just want to make sure you guys understand that. Because that will, okay, let's pause that. We'll pause. I want to make sure this is understood in reference to the following. So let's say this is 100% your stock, right? Concentration. The concentration normally you're given, so just bear in mind, let's say it's 10 grams per liter. Then you're going to dilute it with 1 mil volume here, assuming that it's fully resuspended, by the way, in 9 mils of your diluted. So it goes from, 10 grams per liter to 1 gram per liter, dilution of 10, right? Or the dilution factor is 10 to the minus 1. Then you repeat that 10 to the minus 1 with 1 mil now taken in to another tube that has 9 mils. And because that 1 mil adds up to, with 9 mils to 10 mils, just like there, you now have another dilution, which is going to be 10 to the minus 2 from the total being 10 to the minus 2, because you have 10 to the minus 1 here, and then you're diluting it by another 10 to the minus 1. So 10 to the minus 1 times 10 to the minus 1 is 10 to the minus 2. Then you repeat that stage-wise repetition, 10 to the minus 3 to 10 to the minus 4 to 10 to the minus 5. But the difference here is that you are now taking 0 0.1 mils, right? So, and 0 0.1 mils in reference to your stock, right? So that's something you want to bear in mind, that because you normally would have done these stage repetitions of just 1 mil, you're now taking 10 times less, right? With your pipette, for example, meaning that it's going to be 10 times less than that dilution shown here. which in this case was 10 to the minus 1. So this becomes 10 to the minus 2. And then this was 10 to the minus 2. So this becomes 10 to the minus 3. 10 to the minus 3 becomes 10 to the minus 4. And so on, meaning that you're doing another dilution by 10 to the minus 1 across from these tubes to these plates. So that helps us, especially when we want to determine the number of CFUs, or original cell densities, where we have, colonies between 30 and 300. Technically, this could work. This was made simpler to, do it between 30 to 300 to derive, accurately the plates that showcase cell densities or viable counts that are within this range. And then thereafter, you would multiply or technically divide by the total final dilution factor. So let's say if I had 252 colonies here, what would be my original cell density? Anyone know? 252. And you could put it in whole numbers, scientific notation. Typically for lab exam, I would recommend scientific notation. But if you want to do both, that'd be amazing. Let's say you have 252. So 252 colonies in a plate with 10 to the minus 4 in dilution. Technically, the tube was 10 to the minus 3, and it went to 10 to the minus 4. What would be your CFU for the original sample?
2.54 times 10 to the 6 CFPU per mils.
That is correct. So does that make sense for everyone, by the way? Even if you're typing it, does that make sense? Give a thumbs up if that makes sense, either in the chat or in the group, in your icons. Okay. Yeah, I know it may sound like very simple, but I bring this up. So actually, I point out for Hajra, it's actually 10 to the plus 6. 10 to the plus 6 for CFUs. Yes, Daniela, you have a question?
Sorry, that was an accident.
No problem, no problem. But just bear in mind that, dilution factors, you're dividing by them, they're negative exponents, meaning that when you essentially multiply, you become positive, meaning that it would be 2.52 times 10 to the positive 6. And that's CFUs per mil. And then if you think about it, and then now, like, let's say to bring it into better application, let's say you're dealing with, and this is something you could expect, right? Let's say you are given like a lab instruction to have only 1 times 10 to the power of 5. So 10 to the power of 5, sorry, 10 to the power of 6. 10 to the power of 6 cells in a given solution for transformation efficiency. The question then is how much volume should you add into that tube, for example? You want to get 10 to the plus 6, sorry. 10 to the plus 6 is what you want to get. How much volume do you need to add? How much volume do you need to get to 10 to the 6 CFUs, referring to colony forming units? Because you could technically be asked, and it's more simpler to say, okay, if I have, let's say, 0.1 mils, then okay, I have 2.52 times 10 to the power of 5 CFUs, right? But the question now is framed in a unique way because this is more applicatory. Like if you're now working in a benchtop in your own lab, if you're, tasked with having the right number of CFUs or number of bacteria for a given exercise, right, for transformation, which is hopefully going to come up in the second following exercise. You wanna think, okay, how much volume do I then need to add? Assuming that you resuspended aseptic technique, right? Considering the techniques and methods I've described earlier, I want you guys to think about what you would expect then for deriving the number of CFUs. Anyone know? sorry, to determine the number of how much volume you should add into like a separate tube to get 10 to 10 times 10 to the power of 6 CFUs, which is like 1 times 10 to the power of 6, just so you know. Maybe to make it easy. If you're calculating, that's okay. Keep going. Think about it like this. If you do the inverse, right? If you're doing essentially, if you're trying to find out the number of like the volume now, right? You would essentially divide your expected or what you aim to have as the number of CFUs, which we know is 10, like I won't say it, but you multiply or you divide the expected number, but by what you have, right? So that would be what we know is 2.52 times 10 to the power of 6. I bring that up because then what you would do is 1 times 10 to the power of 6 divided by 2.52 times 10 to the power of 6 is essentially 1 divided by 2.52, right? Because 10 to the power of 6 gets canceled out from the numerator and denominator, meaning that you'll likely get, and this is by the way in mils, 0.38 mils, right? So that highlights to us, okay, then if I'm doing this experiment, I'm going to take 380. I'm just giving an average. The value may be a bit different. Maybe I should show you guys just so we're on the same track. I open up just to get like more precise reading. Okay, I was actually hoping it would be like this. So roughly 0.397 microliters. Sorry, 0.397 milliliters, which is 397 or 396.8 microliters. So you want to, like, that's what your answer would be, 396.8 microliters or 0.397 milliliters, right? So you want to know that level of specificity and that application of the material in this regard. And that's something you could expect in terms of difficulty. Now, when it comes to the plaque assay, we went through this with the FLR. I have a bit of it about it in the manual as well, so hopefully that makes sense. But it's really just applying the material now from viable counts of bacteria to plaques, right? Now to essentially counting the number of plaques that you see on your plates to derive the original population density of your bacteriophage, which is shown as PFUs per mil. I think for exercise 3, I want to show it to you in the manual. Or do I have time to? Let's see. Okay, we have transformation and conjugation. Let's go to the manual. Because these are a bit more lengthy in understanding. And I went over a bit about this separate point, but some of the different conditions, which we could go back to if needed. But streak plating, we talked about. Original cell density, we just spoke about for bacteriophages. Pure culture streak plating. This is an annotation I wanted to try to show you guys. And these are some components that I wanted you guys to talk about for your first FLR, which is great, right? Very happy that you guys did this. And some of you also referred to Dr. Brunt's questions in the outline for the FLR. So that's also great as well. It's a bit of an extension, but you guys could also consider for future labs to include figures. You can ask your TAs if it's allowed, because it would be very beneficial, like in your intro, to include like a figure, let's say of the lytic cycle, or what your bacteria looks like under the scope. That's a separate topic, but just a separate tidbit actually. So, but we'll go forward. I highlighted here for like the plaque assay, bearing in mind the volumes, right? Which we've touched upon before for your FLR. And we'll just go down. That was actually what I wanted to highlight for you guys. Pretty much like the steps of diluting for serial dilutions, your original bacteriophage, which is what you were trying to derive for your FLR when you started off with tube 2, which was at a dilution factor of 10 to the minus 3, technically. And so you would have continued on with the other tubes. You would have had tube 1 as your control, i.e. without the bacteriophage included, but just essentially with water, and then 300 microliters across the board for E. coli. And then, so it's a bit inverse, but the idea is that you want to understand that, just like for viable counts, in this regard, you're doing plaque counts to derive the number of PFUs after you would have, inoculated and essentially incubated a tube of E. coli with your bacteriophage and soft agar, rubbed it around, made a smooth swirl into your plate, then incubated them, not inverted, but incubated as such, and then thereafter, with the NYZ plates, you would have derived plaques the following week, right? And then, so that actually moves us to transformation, because it's another set of apparatuses that involve bacterial manipulation, because you have essentially bacteria that you would think about it like for application. Think about, in your future careers, let's say medicine or research and academia. Think about, let's say you wanted to purify or actually grow a particular protein or gene of interest with expression. There are different steps that we take at the microbiological level or at the biological level to analyze and essentially determine or to culture and to extract proteins or genes of interest. Proteins when you're extracting, and genes for expression. But let's see, if we look down here, bacterial cells you can make competent in two ways. One is highlighted here with calcium chloride and then heat shock. The other is electrophoresis, in a way that you actually use electron voltages to essentially prematurely zap, to penetrate the cell walls, to let them open up porously, to allow genes or DNA to enter into your bacteria of interest. The idea is that you have bacteria of interest and you have genes of interest found within your plasmid. And your plasmid is engineered to be in a vector, and that vector is shown on the following slide. But the idea is that you have genes in like a sequence, like in an assembly, and that is called your operon. You maybe remember this from Bio B12. I remember this with lac operons. And then you probably would have done a similar thing now here with another sugar, and this metabolite is arabinose, right? So that's where the ARA operon, or the operon that we are dealing with for transformation, comes in, because essentially to make it active, i.e. the sequence of events, be it for an original operon or the modified that we used with GFP as downstream from your promoter, from the regulator, the idea is that you would have essentially used the arabinose as a sugar found in our plates to essentially activate, the operon itself, to activate the regulator to allow for gene, essentially to promote gene expression, or I want to put a more particular stance on this. So essentially, as you see here, it binds to the regulator, right? The arabinose itself directly. And then, yes, there is as you can see, a change in shape, right? A conformational change in the sense that promotes the binding of RNA polymerase to promote transcription of your genes of interest, which in our case, just like how it would be in the natural environment of bacteria B, A, and D, in our case, for the modified gene of interest or modified operon, you have the expression of GFP. And that expression leads to the production of GFP as a protein that fluoresces following UV luminescence. So that's where bioluminescence comes in. And that's where, the extraction even of the gene itself, or the extraction of the protein, which actually was from jellyfishes, from jellies, which you see here, comes into essentially the purification, the expression, eventually as genes, that thereby are used to this very day. So, from under the sea to inside the lab, you can see quite a comparative difference and unique application of such similar traits and fluorescence. So think about it like this. You have the fluorescence once again, but the unique component is that it's in a vector that is, genetically engineered or biologically engineered. So you have like this vector that is essentially your plasmid, and that plasmid includes this sequence that you just saw on the previous page, right? From the arabinose, essentially regulatory gene, to essentially the GFP shown here. But and it includes the promoter, which is why I touched upon it. But you have two other components. You have the origin of replication, I've highlighted here, and you have the beta-lactamase gene. Before I go there now, though, you have the vector, and the reason why you need it to be essentially as such, right, rather than just like a single strand of DNA, for example, is because you want to actually have it reproduced independently in your bacteria. And so that's why you're going to transform the bacteria, so that way it could essentially, just like how stains pass through, right, your bacteria of interest. You want genes now to pass through, or plasmids in this case as vectors, to essentially pass through your bacteria to thereby grow, or at least be transcribed and synthesized as proteins and then reproduced in the growing and binary, like in bacteria that would undergo binary fission. So essentially, think about the application of transformation, not just as like a theoretical, like as a concept, but indeed for future operations, right? So that could be including expression of your gene and thereby replication in other, future progeny of bacteria. From there, and we'll talk about that maybe afterwards, but you could see many potential applications because bacteria can grow, as we know, from 10 to the power of 6, 10 to the power of 7, 10 to the power of 8, right, with our CFUs. So you can hopefully see and appreciate the integrative components that are elaborated here. Thereafter, so yeah, I think this is where, let me go. So yeah, pretty much I talked about like, actually any questions before I go forward from anyone? Keep writing them. I'll actually go forward to talking about the origin of replication, because if you recall, maybe lecture material connects with this. Origin of replication is important for bacterial gene expression and replication, right? So you want to bear in mind that as bacteria fizz or separate from one another, which is opposite of fusion, right? Binary fission. The idea is that generally speaking, we have now the replication of genes occurring from the ORI, right, of plasmids, which in this case would be already present in your vector that you've engineered. So that way, you can help the bacteria to essentially replicate and express and replicate your bacteria, sorry, your genes of interest into proteins, right? So, sorry, I'm just moving a bit, but you can hopefully see that you have the given structures. These were included in your operon, right? And the components that are highlighted here for your vector are also important, right? I mentioned about this for ORI, but for B subtilis, sorry, for beta lactamase, this gene is, while really comparing and connecting the dots with our future practicals or our future weeks ahead, but also in terms of its function here, serves important because of the selectable nature of the dye itself. Or sorry, I'm looking at the dyes of the highlight. But essentially, the gene itself serves important as a selectable marker, right? Because as you recall, following the addition of arabinose now, right, to promote gene expression of the operon and the entirety of the vector itself, the idea is that beta lactamase will be selectable to indicate viable colonies that have successfully been transformed with a full vector. Some cases in like outside of this lab, you may have the case of essentially bacteria, like in D21, that you inshallah will see that colonies could be transformed, but with empty vectors, and then some with full vectors. So in our case, you're already starting with a full vector. But in other labs, you may actually see the case where you have to transform bacteria, starting off with an empty vector, to thereby adding in primers, to thereby so you're literally, starting off with a simple plasmid, and then you're reconstructing, you're literally genetically engineering, or like you're modifying the plasmid itself to have the specific genes of interest. And that actually connects a lot to research labs and even to, postgrad, perhaps those that are interested in research, you'll actually be likely doing these steps in your own cells of interest, right? Outside of bacteria, even perhaps. But the idea is that you want to understand how this relates and integrates to the concepts that I've described, because this selectable trait thereafter could be used in other ways. And then thereafter, when you grow and hopefully, can compare these results in the different media that are shown, you'll see that, for example, with the growth of bacteria in the LBA lamp, ampicillin plus a DNA plate, that you would know that these colonies are transformed, right? Because they would, if they were not transformed, right, they would not be able to grow against ampicillin, right? They have beta-lactamase, so they're able to degrade the beta-lactam ring of penicillin based antibiotics, which includes ampicillin and others like amoxicillin, azithromycin, etc. So you want to think about connecting the dots, right? We talked about this in the antimicrobial susceptibility test presentation that you guys had. Hopefully, inshallah, you could see the connections here as well. So you now, considering that, you likely under UV, let me ask you guys, would you see what would you see actually for colonies for the third set of plates? Like the third plate here. Would you see green colonies, yellow colonies, clear colonies, no colonies? Why or why not?
Wait, could you repeat that question?
What do you expect to see in the third, and actually we'll add in the fourth plate? Write it down. I would recommend writing it, Garvin, if you can, because I want everyone to write it down, and we'll garner the thoughts together. Essentially, do you expect to see colonies, yes or no? And what color of colonies would you see under UV? White, green, yellow, and then if you actually expect to see no colonies in either of those two plates, why or why not? I like everyone to write it down in the chat, actually, and I'll give about 2 minutes. Let me just see if we have time. I think we have to go through conjugation briefly, so that'll be the semi-last one. Maybe I'll give you guys a minute if that's okay. Or if you need me to, I'll share it. Okay. So we have no colonies, no color. Is that for the LB amp or the LB amp era plate? I think that's from Alta, the LB amp plate. And then for Alicia, she's mentioning clear colony for 3, and then yellow for yellow colony with green under UV light. Okay, I understand that. And that's for the 4th plate she's saying. And then colonies for both white in white for amp under UV, and then the 4th plate being green from Diane. And then we're seeing from Fatima a very similar component as well. So I would say the last set of 2 responses are correct. And Garvin, were you about to say the same or no?
No, I agree.
Yeah. Reason I'm saying this, because something to bear in mind is that your bacteria, you do want to know, by the way, what bacteria you are using. If it was Micrococcus luteus, technically, yes, it would be yellow to start off with if you see colonies, right? which would technically not be in these two because they weren't transformed with a bacteria. They weren't transformed with a PGLO plasmid that has ampicillin resistance, meaning that these two should not have colonies generally, unless they were transformed in another way, or they actually had resistance to begin with, which is a separate topic to explore. But then your third plate should show white or E. coli looking bacteria, which are generally whitish in hue. And then with arabinose, even with UV, they would remain like white. Technically you want to give a white description rather than colorless. But I understand what you guys are saying, maybe translucent, or we'll say white or opaque. And then when it comes to arabinose being added into that plate, that's where the entirety of the, sorry, I'm just thinking something here. Yeah. Let me just make sure. Okay. So essentially these now transformed colonies for the last plate would fluoresce following the activation or the expression of the PGLO plasmid, meaning that essentially GFP would fluoresce, and then essentially you would have, continuing from there on, you would have essentially, what do we call it? Yes, GFP being synthesized and the protein being produced. Bear in mind that ampicillin, the BLA gene, is actually run independently. So that's actually how you're still able to see colonies, right? Even though you don't have arabinose present. So just bear that in mind. The BLA beta-lactamase gene is still being expressed, but the arabinose, the one that you're using for the operon itself, that's where GFP is being exclusively derived. So just bear in mind, they work differently, independently or in tandem from one another. Now, for conjugation, it's a bit more easier to understand from the presentation, but it's simple once again, actually. We started off with, separate slants of bacteria, HT99 and J53R. I believe they were actually liquid. And then you would have just used the cotton swabs, 1-drop spreads on each side for different plates with separate antibiotics, chloramphenicol, which HT99, and then rifampicin, which is another antibiotic for your second plate. Then after that, you would have done, inverted, place them into incubator, inshallah, and then you would have compared with incubation about 2 hours later, with the right volumes, bear in mind, and resuspension done well. You would have essentially incubated 2 hours, resuspended, and then placed 1 drop into the entirety of this plate. And the benefit of, or the purpose of doing this is to actually note qualitatively the success of conjugation in this regard, right? We have HT99 and J53R. HT99 was your donor and J53R was your recipient, right? And this was of the plasmid to essentially have chloramphenicol essentially pass, which you see as their F plasmid, the gene to pass CHL into your recipient. And students have done this in the past as their FLR, but the idea is that you would have eventually seen that if it was successful with conjugation, the success of growth of your J53R conjugated bacteria. If not, then the concerns could be of, LROD, aseptic technique, LROD burning through, or even another point is the volumes, as I mentioned, right? You need to have the right volumes so that way, rather that the ratios are accurate so that there is a sufficient number of recipients for a single donor to be conjugated with, or to have their plasmids conjugate and pass through this bridge shown as the PLIS. I hope that makes sense. Are there any questions? No, that was a bit quick for the end, but think about these concepts, think about the limitations, know the difference between the donor and the recipient as highlighted here. right? HT99 is the donor for with chloramphenicol. You need to also bear in mind, where is that gene of interest in? Is it in, the bacterial gene itself, or is it in a conjugated plasmid? That's important because if it's in a plasmid, conjugation can occur. But if it's in the bacterial gene itself, then it won't be able to readily pass through because it's technically part of the bacteria itself in its own gene machinery or gene transcription that exists. And so that's something you want to keep in mind. Whereas in the case of rimpycin, it's actually in the bacterial chromosome, meaning that it wouldn't be able to reverse the step of, having J53R as the donor for rapamycin, now placing it into HT99. So bear in mind those details and also knowing, oxytrophic, meaning that they have specific growth factor conditions, right? J53R is limited. As you can see, the J and R, if you know a bit about biochem, you have essentially, actually it's not highlighted here, so it's a bit different in that regard, but you essentially could have certain changes in amino acid constituents that are present in the bacteria. And in this case, at least, it requires methionine and proline to be able to grow successfully. So you would want to bear that in mind when you make plates, right? With the right conditions in place. That concludes lab 4, and now we're into week 5. I want to try to do this in the next, let's say, 14 minutes, inshallah, maybe we'll say 23 at max. And this is relatively simple. And I'm going to go through the manual just to highlight the different stages that you see here, but paying close attention to that's that which is highlighted. Sorry, folks. So remember, if you think about bacteria now, right? We've looked at staining techniques. We looked at many other properties and different, counting methods. Actually, we'll talk about them even more now here. But when we look at generation time, think about how quickly bacteria are able to reproduce, or, in this case, binary fizz from one another. And that's where you have at around 20 minutes for E. coli at the right temperatures at around 20, sorry, around 37 degrees is where you actually have such a short period of time where bacteria are able to rapidly grow or at least for E. coli. And this is a short period, bear in mind, of about 3 hours. And normally we're looking at log phase, but you have other phases that exist that indicate the amelioration or the attenuation of your bacteria in a specific condition. What I mean is that imagine it's like you going to a new home and you're getting attenuated, acclimated to the environment, or you're going up a mountain, right? In this case, bacteria are literally going from one plate to another, or one tube to another. They need to get acclimated if they are aiming to grow, right? So the idea is that specific phases exist, and you have a lag phase where essentially there's intense metabolic activity shown because essentially they're trying to make up, or they're trying to essentially, well, to make it even easier, they're getting attenuated to their environment, trying to see what they can best extract and to best mobilize in order to grow. Thereafter, you have logarithmic phase, where essentially that growth becomes in terms of replication. And so binary fission goes up, up the roof or up in log, which is, as you can imagine, exponential, which you want to keep in mind, right? As we talked about from before for generation time. And then that slowly dwindles, right? You can imagine this is also in log, so this is actually a sharp edge where a stationary phase exists, but essentially it would be more like a plateau, right? So kind of like a plateau that goes a bit up, goes a bit down, and essentially eventually it dwindles to a stagnant point, where essentially you have both growth and death existing, coexisting in that specific location or that condition of bacteria, let's say in a specific media or plate or tube, etc. And then eventually, with so many waste products, and that's something to keep in mind, that there's a beginning and an end for every for creation, like creatures themselves. But in this case, that you have death or at least the growth rate that declines quite readily or quite steadily, considering the sorry that essentially that the cell death exceeds the rate of division. And you have two methods of being able to derive, that change in growth, which is typically shown as a generation time. And I want you guys to understand this from the manual because it hopefully makes more sense in discriminating the two methods. You have total count and turbidity. Total count, which could be subdivided as a direct count to direct count. So direct count, if you recall, was like, let me just make sure you guys understand this component. It goes down to, I believe here, yes. So direct count is actually where some of you were maybe not able to do this, but essentially using the Petrov-Hausser grids. Petrov-Hausser grids where you're able to actually see the number of viable cells. That's like a direct count method because you're literally counting cells. But actually, let me correct myself to let you know how you're able to, determine if it's a direct method or indirect. Before I go there, you have within this total count category, you have the direct count I just showed you using a microscope where you're counting the number of cells. You have viable count where you're actually growing the cells on plates, right? Which we've done and described many times or manyfold. And the idea is that those cells produce a single colony, right? Which is why I'm distinguishing CFUs, colony forming units, from colonies, right? Because you're seeing the colonies themselves, but the units you're seeing abstractly as your calculation to derive the original cell density. So just bear that in mind. It's a bit of a technical, just an impartial note, but essentially you have these viable cells that are growing. And the difference from, even though this is a bit like, indeed, you have to wait for a bit of time for at least the viable counts, because the direct counts you could take directly, right, from, your stock or your growing culture of E. coli, for our example, for calculating generation time. In the case for viable counts, as you recall, we normally see it the following week, right? So that, in a way, if you wanted to do, let's say, an immediate test, right? Just like how we've done for before in determining viable counts for, like, as I mentioned, trying to get like 0.3 or 0.4 mils of a particular culture to do further same day operations. And this would be quite difficult using the viable count method. So bear that in mind. But there are other techniques at play. You can use direct count, as I talked about, but you can also do what we've touched upon more in the beginning of the week, is use turbidity. And turbidity serves as an indirect method because it's not only, see, the thing is, what distinguishes direct from indirect is what you're measuring. You typically want to be, in a direct manner, count or derive the number of cells, right? Whereas, and even though this is a direct method, viable counts, you're counting the number of cells, it is impractical, in the sense that, it would take a longer period of time to actually see from the time that you actually started to plate to the time that you incubated, actually are able to calculate or derive the number of cells or CFUs in your stock culture. The idea is that for turbidity, however, you could do it on the same day. You normally are doing it, as we recall, like maybe at least with the microplate reader, or even let's say back in the day, which was done, you would do it every like 5-10 minutes, repetitions of essentially determining the turbidity. And this is a more indirect method because it counts essentially, or it accounts for the absorbance, right? And this is something that you likely learned from the first week of lecture material, understanding the importance of absorbance, turbidity, the relationships between one another, and essentially being able to thus understand how you are measuring cell density with turbidity. The absorbance that you have versus the sorry, there was another parameter I may have just missed. You have turbidity, absorbance, and essentially there was another one, refraction, transformance. But nonetheless, like essentially you have the absorbance, and it's inversely related to essentially the release or the amount of rays that pass through your bacteria or your solvent, right? So actually, let's go back a bit. Recall that when you're putting your cuvette inside, right, for a specific time point, for example, of a specific bacteria, then you know that you're gonna have light scattering, right? Because UV, or at least actually, I believe this is visible light. That is because it's at the range of 500, our case 550 nanometers. You're going essentially through the cuvette's stationary, or essentially the light is passing through your cuvette, and whatever is absorbed is essentially going to stay there. Whatever is gonna like within your solvent, right? I'm saying absorbed in a more uh, theoretical sense, but whatever passes through is what's going to be read as being subtracted for turbidity readings. So the idea is that you start off whenever you place in and you blank accordingly the spectrophotometer, right? But the idea is that you're doing like the light scatters. But when you're doing the cuvette, when you're like to be able to absorb or to be able to determine the turbidity, what's occurring or what's taken into account is the amount that passes through, i.e. the waves of light that pass through your cuvette. And that is that derives therein the there's another word, intensity, absorbance, one word that just if anyone recalls the word, let me know. There's a word that I wanted to say. It's like the opposite of okay, actually I think I know it. So the idea is like essentially you're passing through with turbidity. I think let's see if it's mentioned. Yeah, sorry, I think yeah. So essentially, yeah, don't worry. The idea is that you have the light, so light scatters, right? But within your cell, like within your cuvette, those light rays that pass through indicate the relative propensity or density of bacteria or of your solute of interest, which is our case, the bacteria, within that cuvette. And that is being used to calibrate or to essentially serve as an approximation of your turbidity, because what passes through should reflect. So the more turbid your cuvette is, the more absorbent it would be. And then so when you're taking the absorbance values, you're essentially going up in the spec 20. You're going up essentially you're increasing in value for your OD readings. And so if we just go down, the idea is that that includes both, bacteria that are living and like that are past. And so that's where essentially the indirect method comes in, because it's actually accounting for absorbance. You're not actually directly counting bacteria. And then secondly, it's actually with the fact that it's sorry, considering that non-living cells are also included, that may overestimate the true propensity of live bacteria or of bacteria that are continuing to grow, right? Or divide by binary fission. So that's something that you want to keep in mind. There's two technical points as to how it's indirect. One, that, it's counting absorbance or measuring absorbance. And then secondly, it's denoting, sorry, yeah. And then it's essentially the absorbance and then the rays of lights. Sorry, something came up. Yeah, so I think that makes sense, I hope, for you guys. But the idea is that you calibrate now your spec 20s to be able to not only blank, right, but to also serve, as we've tried to do, with either viable or the direct counts, to actually have an approximation, like a calibration between the two. Hypothetically, let's say, like if you go up from 0.5 to 0.7 and or 0.8 in calibration, sorry, in your spec 20, you want to be able to have like a ratio or like a correspondence that reflects, what your turbidity is and how that relates to your viable count readings. Um, as turbidity, so I know it sounds a bit confusing, but as turbidity increases, so as it gets more cloudy, absorbance should increase. Absorbance now, okay, let me try to give you an understanding of this. Yeah, like uh see, technically what's passing through, so maybe to make it simpler, you're considering the cuvette as your mode of measuring absorbance, right? So if it's more cloudy, it's likely going to absorb more of the UV, and that's what you're going to see as your OD readings. Technically, there's other processes that go through, but to make it simpler, increase in turbidity, increase in OD. And that's where you see it often get more cloudy over time, especially after as we were seeing from 0 to 3 hours from the lab manual, you'll see like the log phase continue. Does that make sense, Daniela, by the way? Absorbance increases. There's another, yeah, so don't worry, just trust me that turbidity and absorbance, they're correlated. But then generation time is something that we were measuring, right? As a time that it takes for, bacteria to double in number, right? Within the log phase or cells in our case. And actually, let me just go back. Sorry, something just came to mind. Never mind. Generation time, though, I hope that makes sense. Um, that's no problem, Carmen. Take care. The other thing, though, just to make sure that you guys are aware, you have essentially uh, generation time being derived in two ways. One that I've asked you guys was using the slope shown here. And then technically, this way also works in a similar route, in that essentially you're doing log y2 minus log y1 divided by essentially, in this case, x2 minus x1. And then that's your slope, right? So you would have plotted your absorbance readings, the viable counts across time, 0 to 30 and every 30 minutes for viable counts, to 90 minutes, to then every 15 minutes from your microplate reader, because it would go up and essentially sequence the absorbance readings at a 15-minute interval. So the increments would go up and the absorbance readings normally would have went up as well. Now, just understand briefly that you use these two formulas to get likely the same reading for your generation time. What I mean is that, okay, you have the mean growth rate constant. This is essentially you taking into account pretty much the entirety. In fact, I believe, let me just see if I can go back because I want to make sure that let me see if I'm not missing anything. Yeah, so it would be every 15 minutes, and you would have the blank, which is normally on this side, to be able to calibrate the values, right? Because your blank would also have an absorbance reading because it also may have solutes, the media itself, right? The different constituents, the nutrients, the metabolites, etc. That serves as your blank, and then your other treatments are like your blank plus your bacteria. So, and then across time. So that's where, the viable plates, that's where your values come in. And actually, maybe I'm just gonna briefly go here. We talked about actually, here we talked about the material, the phases I have mentioned thus far. There was one thing, yeah, transmittance was the word I wanted to talk about. So transmittance is the opposite, right? Absorbance and transmittance are inversely related, right? So the more that cuvette absorbs, the more bacteria that you have, the less is transmitted through your spec 20, right? And then so just understand that when you're doing absorbance readings, it reflects inversely transmittance, but also it reflects the propensity of growth of bacteria within that cuvette. And so that hopefully makes sense. That more cloudy, like essentially that's pretty much what it means. We talked about this. We'll talk about this maybe in week 6 or like inshallah in our next review. But definitely, these are things that you want to consider. The other conditions that essentially, as I mentioned, for growth can be shown in log phase, right? Which we show actually in the lines shown here in semilog with the logarithmic increments shown here, 10 to the minus 1, let's say, to 10 to the 0. Well, actually, we did it for absorbance readings less than that, 10 to the minus 2, 10 to the minus 1, 10 to the 0, which is like 0 0.01, 0 0.1, and then 1. So essentially, we took those values into account, and then arithmetically, you'll find that these values are essentially exponential. And then Petrov foster grid, I'm going to talk about the red counts at the end. And then these were questions that you guys wanted to actually answer, if you recall, with question 3 referring to the direct counts using the dye, for example, and then the plaque assay, the results that you saw, right? You expect this, by the way, to be, it's also shown, but you would expect this to be the lamp plus era LB plus LB plate, right? As you could see, that the GFP is fluorescing the bacterial transformed colonies. So that's something that you want to definitely consider. Any concerns, limitations, thoughts of contemplation. Before I end off, so let's go straight down, in fact. Yes, so we use 550. Actually, are there any questions as I go down? You would have done serial dilutions for the viable counts, and then like when you grew those plates thereafter the following week, we would have counted 30 to 300, deriving from the plates that you did with reps of the serial dilutions, which ones or one fits within that range, and then derived the viable, the total number of CFUs per plate, and then use that to actually derive the changes across time to determine the GT, which so I mentioned this before. There are 2 technical paths, and the same exists with the microplate reader, just with the blank here, the different groups. So this would have been the blank, and then the different time points, which I would have done manually I don't know if I did it manually, I forgot. But nonetheless, oh, I think there were actually 3, yes, I recall. So these were reps, right? So we would have done them, the 3 reps, right? If you recall from the guide as well. Hopefully that would have, hopefully this making sense. For each of the groups, you would have had reps, and we would have seen the changes of absorbance over time. And then, so imagine you would have plotted those values, right? And you would have done this on the semi-log paper that you saw on the slide before. And this hopefully gives us a bit of an understanding as to what you would have done thereafter with the tables, the title, like with the points plotted out, right, for absorbance and viable counts. You would have essentially plotted them accordingly across time, bearing in mind the logarithmic scale, right? So like for viable counts, this could be 0. To start off 0.01, and then 2 could have been 0.1, and then 3 could have been 1. And then so you would have artificially looked through the different boxes, the number of boxes in each large increment, to derive the accurate, values to essentially plot or to plot accurately and reliably the different y increments or y coordinates across the different x coordinates for time in both assays of microplate and viable counts. You technically could also do this with direct counts, but that would have been a bit more laborious. But the benefit of that is that you don't have to wait overnight or the following week even to derive, viable counts, because the direct counts serve a similar path, which essentially, if I go to now, you would have actually come to appreciate, because, for example, you would have counted, let's say, 16, cells, right? 16 bacterial colonies within, let's say, a set of 16 squares. And then you had the dilution factor, which you would know, right? Based on how much you diluted your bacteria. And then the volume of each cell, which actually is mentioned, yes, in the lab manual, is 5 times 10 to the power of negative 8. So you would have just essentially done the calculations for each OCD, which is a quite readable way, like within 30 minutes or less, maybe even, maybe even 10 minutes or less, you can actually derive maybe 8 minutes, like the CFUs or the OCD or original cell density, which accounts for these, which accounts for the dilution factor and essentially what you would have mimicked thereafter for viable counts. But that's apart from that, you essentially would have done this to determine, the generation time, and then from there on, you would have used what I've mentioned from the guide, from across the different, techniques. You would have essentially plotted within that line of best fit. You would have taken 2 points within that line of best fit, not just from your, your points indeed, your raw points that are corrected with the mean and from the blank itself. But you would have hopefully, inshallah, actually taken in the plots, the line itself, to take out points to determine the slope. The slope was shown here, right? What I mentioned earlier, and then you would have divided by technically ln 2, which is a log of 2, which is 0.301 or 0.3 divided by the slope. That setup is pretty much the same as the one shown in the manual with that longer equation, like k equals log n2 minus log n1, which is like your y2 minus y1, divided by t, which is technically x2 minus x1. And then pretty much beside it was 0.3, 0.301, which is essentially you doing the entire GT like calculation altogether. Pretty much it would have been the exact same, minus the fact that you technically have 0.301 here, because that would have reflected the log calculation. So essentially, that was that single step. There is the other point of you actually, yeah, I think that makes sense. Hopefully, that pretty much hopefully summarizes the entirety, I hope, of the lab, the weeks 1 to 5 in a nutshell. Are there questions, though? Anyone? There are questions, like critical questions here, that you may want to consider incorporating for your understanding of the material, right? Like based on the conditions, like if you change temperatures, that was a question mentioned afterwards, or if you use a different form of media, this minimal salts media would, even though it has glucose, not be as high, rich, and medium like the LB, Maria Bertani broth. So bear in mind that, differences in media can impact differences in metabolism, differences in proliferation, differences in binary fission, and thus could, in this case, reduce perhaps regeneration time, or sorry, impact regeneration time, making it longer, i.e. larger, meaning that it'll take more time to actually, proliferate or divide. Or if it's shorter, i.e. that it's in more optimal conditions, or it's, going through the log phase quite rapidly, then essentially, you're essentially increasing in binary fission in more optimal conditions. So think about those different components, and hopefully, inshallah, for next week, we'll come to appreciate the other conditions that exist, be it environmental, be it chemical, or even like in the case of physical UV exposure. So think about those 3 different components, what we described, what we've compared. Inshallah, I hope you guys will be able to understand the material quite readily in a different, different conditions that existed as well for the other tests. Any questions? How was that for a bit of a rundown?
Good morning. I wish that we had this Zoom meeting before our log graph assignment.
Which one?
The review for the log graph assignment. I wish we had a Zoom meeting.
Oh, I would have loved to. I know it's so well. I hope I went through it though. Well, I hope I did in the many week 5 discussions that we had for understanding generation time. Remind me, Alta, you're from practical 8, right? Or practical 2?
Practical 8.
Practical 8. So I recall we had, so this is a separate component, but the idea is that I wanted to make sure that you guys could be well prepped. So I took, what do you call it? I did a recording, I think, but nonetheless, don't worry. Hopefully this will help, or at least if you have more, if there are questions regarding this, and even for the other FLR components, please let me know. So I would love to continue supporting you guys in every way, shape, or form. Maybe I'll go through some common questions, or some common comments. And when I mean questions that maybe you guys are thinking. So when it comes to calibrating your ocular micrometer, right? To start off, remember that this is what you're seeing under under your eye microscope. You're seeing this ruler, right? And you're seeing typically an object, right? Like a cell, a capsule, an organelle, etc. So, and even like in AO2, maybe like the epidermis, which is a whole plant cell layer of cells. So the idea is that you essentially are seeing these OUs, right? And you need to calibrate those OUs because essentially you don't know how much each OU is at that given magnification, which is something you want to bear in mind and account for. So essentially you use a stage micrometer, right? As shown here. And you know the values that depict those small increments as being 10 microns or essentially 0.01 millimeters. And then the larger ones refer to, 100 microns or 0.1 millimeters. And so you would take that into account. You would overlap the two rulers, right? Think of it like, back in the day, calculus or high school math. So you overlap the two to derive, okay, what the relationship is, right? Let's say 7 OUs equals 100 microns. Or even 8, let's say. I'm trying to see if they can actually overlap. Yeah, maybe. Maybe we'll say 8. We'll say 8. 8 OUs. 8 OUs equal 100 microns. So 100 microns divided by 8 would give you about 12.5 microns. So now if I look at this, object, so now that you've done that calibration, you now have to look back at your object. Maybe that's a question that you may ask. Oh, how large or how big or how long is that object from this plane or this axis of that I'm viewing the object in my field of view? So it's maybe 5.5, let's say 5.5-ish OUs. So 5.5 times 12.5, maybe will give you probably 66 microns, right? 67 maybe or 68. So the idea is that you now are able to see in practice, now that you've observed that organism, you now can appreciate, wow, this is actually 68 microns. And if you were to change up the mag and you were to look even more detailed, you may be able to get more precise readings, right? Could maybe not only be let's say, you know, from a range of 65 to 70 microns, maybe it could be 68 at 10x objective lens. Then from 68 to 40 at a 40x objective lens, we now realize it's like maybe 68.6 or 68. Actually, no. Yeah, or like like the idea is like you could go from like larger increments being like unable to derive, let's say, the difference between 10 microns to, you know, with resolution, increasing resolution from that. And that refers to the definition being able to distinguish two objects that are closely aligned or closely or in close proximity to another. As you go down in mag, as you increase in magnification, you're increasing in resolution. So you're able to hopefully appreciate smaller and finer details, you know, with the microscope that you're using. And then the oil immersion helps even better to solidify that. You have an oxochrome, and even though it's, I didn't mention it, simple staining, the difference between a chromophore or, yeah, actually, pretty much you have two components of a chromogen. The chromophore makes the color. The oxochrome has a charged component, like crystal violet has a violet chromophore, but it's positive as an oxochrome. So that's something you want to bear in mind. Different, you know, dyes may be charged or uncharged, and that connects also to biochem. So hopefully you guys can appreciate the connections. And then you have different, you know, some are, if they're basic, some are like, you know, some are basic and positively charged with as oxochromes. So if they're like plus like crystal violet, then they're basic. Acidic, they are negatively charged. And you can see and appreciate the impacts that they have with heat fixation as well. Bear it in mind. So no problems, no problem. Sorry I'm saying this out loud, but no problem. Take care, Julia. Have an amazing day. Let me know if you guys have questions. And then I know I'm going over it more in depth, but I would really appreciate you guys to feel free to share questions to incorporate that level of discussion. So I know it's hard to do it now, but hopefully, like, think about how you can incorporate these different topics to other concepts that we'll be talking about in our next review, and hopefully, inshallah, for your understanding in the lab exam itself. But if everyone wishes to leave, they can. If you have questions, feel free to ask, and I'll just skim through the rest of the highlighted components in case I've missed anything. Yeah, the mordant I mentioned stains or adheres to, like in the case of crystal violet with gram staining, that helps to connect the iodine with the crystal violet. It's complex. In this case, the flagellar stain, even though we didn't do it, it actually adheres to layers of the flagella, which you could see with different categories or classifications here based on the images highlighted on the right. And we had like, and I really wanted to touch upon applications. So really try to understand the applications, be it of the different bacteria that we've observed, different stains and their connections to microscopy, molecular biology, medicine, research, etc. A lot of this is quite applicable, even though I know it's timed and it's a bit anxious perhaps for many of us, but understand, like, you know, there are different characteristics and different properties of bacteria, not only in like stains, but in morphologies, in what they include, in capsules, in, you know, in endospores, and even their motility, right? Some are able to move, some aren't. Some are flagellar, some are not, and some have multiple flagella. So those are components that you want to think about. And I mentioned about the mordant as well, for grams iodine. But nonetheless, yeah, just bear in mind like the different setups, the different techniques. We've touched upon a lot of introductory material from the net. And I do want to highlight that there are, as we've talked about certain, you know, expectations, think about the limitations as well, as I mentioned here, for gram variability. Sometimes you may have made them either too thick, or if they're too thin, then it could also give you gram false negatives, right? because they could have been more easily decolorized, right? So it would almost assume that you're doing over decolorization, though typically not the case. But the idea is that you want to bear in the age, the staining, age of your bacteria, the staining quality, the smearing quality. Like if you, for staining, over decolorized, under decolorized, if you overstained, understained with either the primary or counter stain. And then like considering those with the implications of what those bacteria have, right? Think about like, oh, B. subtilis, it's an endospore forming bacteria. Connect how it, is resistant in certain conditions based on the conditions you found it in your tube in week 1, all the way to where you're using it in week 10, and hopefully being able to see connections there, or even of Staphylococcus epidermidis, you're in week 2 all the way to week 10 with biofilms. So like a lot of connective and interesting material. And really with the limitations, once again, like you can have over decolorization, under decolorization. If you forgot, maybe you're missing certain steps, be of the decolorizer or of the mordant, and those can also impact your results accordingly, for either having false negatives or false positive results of gram variability, or false gram positives, false gram negatives. So false gram positive means like they appear gram positive, but they are not. So they're false. Or like false, like giving you an indication that they're not actually that specific category. But they appear to be that essentially. So, and then you have controls, right? Like with your unknowns, you want to make sure that, oh, like, and you should be aware, let's say, Lactococcus lactis, for example, right? If it's, by the way, this also connects us to the Snyder test, right? Because of the bacteria that we're using, lactobacilli, as in, even though this is Lactococcus, but technically it also would ferment, or look at, let's say, let's say this was Lactobacillus, or let's say, let's say it was Lactobacillus lactis. Be very similar indeed. But you just want to bear in mind like the importance of knowing certain controls. And if your controls did not work out and they didn't have the right results, then what could have been done? What wrong? Like what steps could have been missed? What steps could have been done inaccurately? And then what you would have done to resolve those steps. And actually, what then can you thereafter conclude from your results? Are we expected to know which bacteria were used for each lab? Not necessarily, but you should know general traits of those bacteria. So for example, and I know that may sound hard right now, but like, okay, let's say I'm looking at L. lactis. I know that it's L, so the L refers to lactococcus, right? So even without looking here, but like, I know it has a coccus in there, right? So meaning that it's a cocci, right? Meaning that it's in circular or spherical shape. Yes, ovoid can be like noted, but generally cocci is something you want to bear in mind. And then I know, right, from unknown and from the flowchart that I did, inshallah, that these are gram-positive bacteria, right? So if I were to put them in a gram stain, they should appear purple, even if this was considered my control, as a positive control. And then like the other features that you may see, like, okay, I know that it's, for example, capsule positive in skim milk only, right? And it's endospore negative. Like generally speaking, if you look back into your flowcharts, you hopefully will be able to understand like what is going on, and you'll know what distinguishes them accordingly. Like some are in clusters, some are positive, gram-positive, negative. That's E. coli, right? Also motility, generally speaking. Um, no problem, no problem. And then spore stain, yeah, let me just see. Yeah, so B. subtilis does have an endospore, whereas Lactococcus lactis has a capsule. So those are the two main ones. And then color, right? Micrococcus roseus, pink, red, and then you have the Micrococcus luteus as yellow. And then endospore stain we talked about a bit. Hopefully you guys can connect the dots and see the importance of certain steps, limitations at play. And then If you keep going down, techniques, right? Methodology, serial dilutions, making sure that you not only are following like the techniques itself, but volumes, for example. You're doing 1 mil and 9 mil inoculations for diluted. It's not like 0.1 mils or 0.8 mils or 0.9 mils, but you're doing 1 mil into those specific dilutions. Because if you underdo it, let's say 0.9 mils, then each of the other ones, if you kept going in that route, let's say 0.9 mils in my first inoculation to 0.9 mils in the following ones, then you will notice that those limitations will be magnified, meaning that these calculations, not just of your final dilution, but even of the colonies themselves, may not be fully reliable unless you account for those differences. So just bear that in mind. You can technically account for them, but you want to make sure that you would have done that from the very beginning, and likely would have stayed with the 1 mil inoculations for each, or 1 mil aliquots for each of the transfers that you would have done. And then for calculation, just bear in mind, some of you may have mistaken this, but your tubes normally are at specific dilution. If you're adding in 0.1 mils, then you're going a further dilution down, just one down from 10 to the minus 3 to 10 to the minus 4. If it's a plate that is going with 1 mil for a particular reason, then it's actually going to stay as the same dilution. No problem, no problem if you have to go. Let me just see if there's any other things. I think we're at, yeah, so we did this nice calculation as well. And then now into week 4, understanding the use of another plating technique, streak plating, where you're able to dilute your bacteria, transforming your bacteria, doing conjugation, and even, plaque assay, inshallah, something that I hope you guys were able to appreciate from the FLR, determining the number of plaques, PFUs, and bearing in mind that you have specific, control that's important, right? To account for a full smear on your plate A, right? And then accounting for the volumes for the other, conditions. Just like for conjugation, there's a specific, ratio of your bacteriophage here with your cells, and conjugation for your donor to your recipient. So you want to bear in mind that you're adding in the right volumes and in the right ratio. So like, for example, here, you could also do it at 200 microliters of the lambda phage with 600 mils of your cells. But then the other, setups would also have to be multiplied in 2. And this is usually if you're doing bulk analysis or mass, mass methods, all like you're doing like let's say a cocktail, or like you're doing a mass mixture of different sets of, like multiple plates, for example, as reps for a specific or multiple exercises. So I talked about this. I went over a bit more into the lab manual here. So I hope, inshallah, many of this are the materials here you would be able to skim or like look back to, importantly, but also account for when you're doing your slide review as well with critical questions. Also, bear in mind, I didn't talk about this, but PGLO was at 10 microliters, right? And you had a concentration of 0.005 micrograms per microliter. And I know this sounds hard, but just understand that, okay, you're starting then with 0.05 micrograms, right? Because it's 10 microliters as a mass. And then you would use the calculation of essentially the total number of transformants. Let's say it's 100 transformants divided by 10 micrograms. That's going to give you essentially for each microgram of plasmid that you add, essentially 10 colonies that are transformed, ideally, inshallah. And that's a good way to understand, I believe, the following question, which I believe is either here, right here, 8D. So hopefully you guys will be able to understand that you had, and you can also account for actually something very important, the ratio that you're also using for transformation. So you plated only 200 microliters, which can happen, right, for both plates, LB amp plus LB amp plus ERA. So those are 2 plates, 200 microliters in total, 100 microliters each. 200 microliters in total, divided by that 510 microliters of this whole solution, is something that you're going to account for when you're essentially deriving the appropriate value. So it technically, even though it's not mentioned here, you're technically multiplying that value. So I said 10 micrograms, right? 10 micrograms or 10 transformants per microgram of PGLO. Let me see if I can actually do this. Yeah, so you could do essentially multiply by 200 divided by 510. And then so essentially you would get roughly, so 2,000 micrograms times microliter divided by 510. That would give you about like four true, like within your sample, essentially. Some people will go with accounting for the volume, and that's if you provide it, so that's to specify, right, the total, like from where you're starting off from. So just bear in mind that you're accounting for the differences. It could also work the other way around. And I said 10 because like I was being very simple, right? Or like very generous, like saying 100 transformants. But bear in mind that could be in like 10 to the power of, -3, right? So for the case of, or like in our case, even if it was 10 to the -2, hypothetically. Oh, this is by the way in micrograms, so that's a whole another, apparatus. So just bear in mind these are the same AD and 8D and 8E, but you just want to bear in the values or compare based on the calculation that you've derived accurately what the efficiency would be. And we talked about, the setup for mechanisms here, but hopefully you can appreciate and look back at the mechanisms of conjugation in greater depth with the pili, death plasmids, and the requirements therein. And then we talked lastly about binary fission every 20 minutes for a short period of about 3 hours, the log phase, the molecular or the microbial analysis there, and then the instrumental analysis of using total count, direct method, turbidity as indirect method, using either direct or the viable counts for the direct method. And then log and then solutions. Any final questions, folks? And then you would have essentially used the microplate, viable counts, would have calculated, derived the values here, and then essentially would have thereafter determined GT, would have answered some of these questions here as well to understand a bit more. Even though I didn't penalize, but understand what I've described, right? Extend the conclusions and the analysis or the results that you found with your interpretations to extensions that exist beyond the lab, to limitations that existed from the lab, to essentially extensions and applications that you could use down outside of the lab yourselves. Oh yeah, and we had the direct count shown here with the calculation of the volume of the space here, 5 times 10 to the power of -8 mils, rather. So not microliters, but mils, and then molecular density, 5 to 15 in each small square. No problem, Daniela. No problem. And everyone else, if you have any final questions, please let me know. But apart from that, I'm looking forward to your success. And finally, please let me know if you have any questions, concerns, thoughts, anything that you want to share. And yes, you can count accordingly. If there's any questions, let me know. Once again, great to have you guys. And سبحانك اللهم وبحمدك نشهد أن لا إله إلا أنت نستغفرك ونتوب إليك. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. والعصر إن الإنسان لفي خسر إلا الذين آمنوا وعملوا الصالحات وتواصوا بالحق وتواصوا بالصبر. That's my concluding statements. So I'll leave you guys. Have an amazing 1 minute early end to the review, and looking forward to seeing you. Assalamu alaikum. Take care.